Find somewhere comfortable tonight. Wherever you are, whatever position your body has settled into, that is exactly right. You do not need to sit up straighter or arrange yourself differently. You do not need to hold on to any of this. The words will come and go like water moving past you in the dark, and that is perfectly fine. If today was long, you can set it down now. If something is still turning over in your mind, some conversation, some worry, some half-finished thought that keeps circling back, you do not have to resolve it tonight. It will still be there in the morning if it needs to be. For now, there is only this, a strange and beautiful story about light. We are going to travel tonight, not anywhere that requires effort, just gently the way the mind moves when it is beginning to release the day. We are going to go down deeper than sunlight reaches into waters so vast and so ancient that the world above them barely seems real. We are going to visit forests at dusk and warm summer fields and the edges of bays where the water itself seems to dream in blue. Because tonight the subject is light, not the harsh, demanding light of screens and overhead bulbs and the sharp glare of afternoon. Something far older than that. Something the living world discovered long before we arrived and has been quietly practicing ever since. Tonight, we explore bioluminescence. Life that makes its own light. You do not need to concentrate. You do not need to remember any of this. Just let the images arrive, soft and unhurried, and let them carry you somewhere still. We are in no rush at all. There is a bay in Puerto Rico, on the southern coast of the island, called Mosquito Bay. It is not a particularly large body of water. On a map, it looks almost incidental, a small curved indent along the shore. But on a moonless night, if you lower your hand into the water and move it slowly, something extraordinary happens. Water glows. Not faintly. Not in a way that requires squinting. It glows with a clear, living blue, the color of something dreamed rather than seen. Every movement you make leaves a trail of light behind it. A fish darting past becomes a streak of cold fire. A kayak paddle lifted from the surface drips luminous drops back into the dark. Children visiting for the first time sometimes go very quiet. Adults too. What they are seeing is not a trick of the light. It is not a reflection of anything above. It is life producing its own illumination from within. Tiny organisms, each one invisible to the naked eye, each one smaller than a grain of sand, responding to disturbance the only way they know how. With light. This is bioluminescence. And it is one of the most widespread phenomena on this planet. It is happening right now, in oceans you will never visit, in forests you will never walk through, in the deep dark places of the world where no human eye is watching. The living world glows, quietly, persistently, for reasons that took us centuries to begin to understand. The ancient Greeks noticed it. Aristotle, writing more than 2,000 years ago, observed that certain things glowed without warmth. Decaying wood. Dead fish left on the shore. He noted the strangeness of it. Light without heat. He called it cold light, and the name has stayed with us because it is still the most accurate description we have. Pliny the Elder, writing in the 1st century, catalogued glowing jellyfish and fireflies and the strange luminescent trails left by ships moving through warm seas at night. He did not know what caused it. Neither did anyone else for a very long time. It was simply one of the world's quiet mysteries, observed and wondered at and not yet explained. The explanation, when it finally began to emerge, turned out to be both elegant and astonishing. And we will arrive there, gently, in our own time. Imagine you are holding a firefly in your cupped hands on a warm evening. The light pulses through your fingers. It is yellow-green, soft, intermittent. It does not burn. You could hold that firefly for hours and never feel warmth from its light. That is not an accident. All bioluminescence works through a chemical reaction. At its heart are two key players, a molecule called luciferin, which is the thing that actually produces the light, and an enzyme called luciferase, which is what makes the reaction happen. The names come from the Latin word for light bearer, and there is something quietly poetic in that. Life naming its own inner illumination after the thing that carries light. The reaction itself is almost impossibly elegant. The luciferase enzyme takes the luciferin molecule and, in the presence of oxygen, causes it to be oxidized. In many organisms, particularly insects, the reaction also requires a molecule called adenosine triphosphate, which is essentially the body's energy currency. As the reaction proceeds, it creates an unstable intermediate molecule, a high-energy compound that cannot hold itself together for long. When it breaks down, the resulting molecule, called oxyluciferin, finds itself in what chemists call an excited state. It has more energy than it can comfortably contain, and so it releases that excess energy the only way available to it, as a photon of light. That is what you see when a firefly pulses in the dark, a molecule relaxing. Energy finding its way out as light rather than heat. The process is so efficient that less than 20% of the energy involved is lost as warmth. Compare that to an ordinary incandescent light bulb, which loses roughly 90% of its energy as heat and only converts 10% into actual light. The firefly is, by any measure, a far better lamp. What is remarkable is that the specific chemistry varies enormously across different species. Fireflies use a form of luciferin called, straightforwardly, firefly luciferin. Many marine creatures use a different molecule called coelenterazine. Bacteria produce their light through a mechanism involving a compound derived from flavin mononucleotide. The same fundamental trick, the same conversion of chemistry into photons, achieved through dozens of different molecular pathways that evolved completely independently of one another. This is one of the most striking things about bioluminescence. It has evolved not once, not twice, but at least 94 separate times across the tree of life. 94 distinct occasions on which some lineage of living things stumbled upon the ability to produce light and found it useful enough to keep. The earliest known example appears to be in ancient marine organisms called octocorals, roughly 540 million years ago. Before complex animals had fully arrived, before the world's oceans looked anything like they do now, light was already being made. That number, 94 independent origins, is worth sitting with for a moment. It suggests that making light is not some extraordinary biological accident. It is something that life discovers over and over again whenever the conditions are right, because it solves so many problems so well. The problems it solves, and the extraordinary variety of solutions different creatures have found, are what we will wander through next. The surface of the ocean is where most of us imagine the sea to be, sunlit, moving, full of color. But the surface is a very thin layer. Below it, the ocean descends for kilometers through a twilight zone where sunlight becomes a faint blue suggestion, and then into absolute dark. No photon from the sun has ever touched the deepest ocean floors. Nothing down there has ever cast a shadow. Yet it glows. An estimated 80% of all deep-sea creatures are bioluminescent. 80%. In a world with no sunlight, in a world where the only light available is the light that living things make themselves, the ability to produce and detect light has become not a novelty, but a fundamental feature of existence. The deep ocean is not dark in the way a closed room is dark. It is dark the way a clear night sky is dark, full of points of light, of moving glows, of creatures signaling and hunting and hiding and finding one another across the cold and the pressure and the vast distances. There is a fish called the anglerfish, and if you have ever seen an image of it, you will not have forgotten. It is not a beautiful creature in the conventional sense. It has a wide, hinged jaw full of translucent teeth, and a body that looks unfinished, rough-edged, designed for function rather than elegance. But projecting from its forehead, on a long, flexible stalk, is a small, glowing lure. A lantern of living light dangling in the darkness like a beacon. The light in that lure does not come from the anglerfish itself. It comes from bacteria. Billions of bioluminescent bacteria living in a small organ at the tip of the lure, fed and sheltered by the fish in exchange for their glow. The anglerfish and the bacteria have an arrangement, ancient and mutually beneficial. The fish provides a home, the bacteria provide light, and small creatures, drawn to that light in the darkness the way moths are drawn to a flame, swim toward what looks like a safe and interesting thing and find instead the anglerfish's waiting mouth. This is one of the most common uses of bioluminescence in the deep ocean. Predatory deception. The production of light not to be seen, but to attract something that should not be seen. But the anglerfish is only one example among thousands. There is a fish called the stoplight loosejaw that produces red light from organs beneath its eyes, a remarkable adaptation because most deep-sea creatures cannot see red light at all. For them, the red beam is invisible. The loosejaw can illuminate its prey without the prey knowing it is being watched, a biological spotlight that only the predator can see. There are creatures called marine hatchetfish that produce light from their undersides, but not to attract prey, to disappear. The faint light filtering down from the distant surface creates a dim glow from above, and anything swimming below a hatchetfish would see its silhouette against that glow, a clear and visible target for a predator. But the hatchetfish produces light from its belly that precisely matches the intensity and color of the light coming from above, cancelling out its own shadow, erasing its silhouette. This is called counter-illumination camouflage. The creature uses light to become invisible. It is one of the more elegant solutions to the problem of being eaten that the natural world has produced. And then there are the brittle stars. Relatives of starfish, slow-moving creatures that pick their way across the ocean floor on 5 long, delicate arms. When a predator seizes one of those arms, the brittle star does something extraordinary. The arm begins to glow brightly and persistently even after it has been detached from the body. The brittle star releases it and retreats into the dark. The predator is left holding a glowing, wriggling arm that distracts it long enough for the rest of the animal to escape. The arm will grow back. The brittle star, for now, is safe. It used light the way a magician uses misdirection. And it worked. Bioluminescence is overwhelmingly a marine phenomenon. The ocean is where it is most common, most varied, most deeply woven into the fabric of life. But it has found its way onto land as well, and in doing so it has given us some of the most familiar and beloved encounters with living light that most of us will ever have. Fireflies are perhaps the most widely known bioluminescent creatures on Earth. They are beetles, and there are roughly 2,000 species of them, distributed across temperate and tropical regions on almost every continent. On warm summer evenings, in meadows and forest edges and suburban gardens, they rise from the grass at dusk and begin their conversations in light. Because that is what the flashing is. Each species has its own pattern, its own rhythm, its own particular sequence of pulses. The male flies and flashes. The female, perched somewhere in the vegetation, watches. If she sees the right pattern, the pattern that belongs to her species, she flashes back. The male follows the response. They find each other in the dark through nothing but the language of light. The synchrony that sometimes develops in large groups of fireflies has a particular quality of strangeness to it. In certain forests in Tennessee and in parts of Southeast Asia, thousands of fireflies will sometimes begin to pulse together, waves of light moving through the trees in coordinated rhythm as though the whole forest is breathing. No one is directing the performance. It emerges from each individual responding to its neighbors, adjusting its timing until the whole forest falls into phase. Watching it, people often go quiet. It has the quality of something that should not be possible, and yet is simply happening, unhurried, in the warm dark. The light a firefly produces comes from an organ in its abdomen called the lantern. The chemistry inside is the luciferin-luciferase reaction, the same fundamental process as in the ocean, arrived at independently, producing light in the yellow-green range, around 560 nanometers. The firefly controls when to flash by, regulating the flow of oxygen to the lantern cells. A pulse of oxygen triggers a pulse of light. The firefly is switching itself on and off like a lamp, using its own breathing as the switch. Away from fields and warm evenings, there is another land-based bioluminescence that most people never think of. Fungi. Certain mushrooms glow, not all of them and not brightly, but in dark forests on dark nights, the fruiting bodies of some fungal species emit a faint, steady, greenish light. There are roughly 100 species known to do this, scattered across different fungal families, suggesting that here too the ability evolved independently multiple times. The glow of bioluminescent fungi is not the intermittent pulse of a firefly. It is continuous, soft, and patient. A decaying log colonized by glowing mycelium will glow all night, every night, for as long as the fungus is active. In older times, this phenomenon was called foxfire or fairy fire, and it was noticed by people walking through forests at night, a faint greenish shimmer coming from the ground or from fallen wood with no obvious source. It contributed, in its quiet way, to the sense that forests at night were inhabited by something beyond ordinary explanation. The function of fungal bioluminescence is still not entirely clear. One compelling hypothesis is that the glow attracts insects at night, and those insects carry fungal spores away on their bodies, helping the fungus disperse. There is something worth lingering on in that idea. A fungus, rooted and still, unable to move, unable to call out, producing light from within itself as a way of saying, quietly and persistently, I am here. The story of how we came to understand bioluminescence is, in its own way, a story about patience and careful attention, about people who noticed something strange and could not stop wondering about it. Robert Boyle, the 17th century chemist, is one of the earliest figures in this story. He placed glowing wood and glowing fish in a vessel and removed the air from it. The light went out. He let the air back in. The light returned. This was a significant observation. Whatever was producing the light, it needed air to do so. It needed, specifically, oxygen. Though that word did not yet exist in the scientific vocabulary of his time. Boyle did not know what oxygen was, but he had identified something essential about the process. The story moves forward to the end of the 19th century and a French pharmacologist named Raphael Dubois. Dubois was studying bioluminescent clams, small creatures found along the Atlantic coast of Europe, and he performed an experiment of quiet elegance. He ground up some of the clams and produced two extracts. One was heated, which would destroy any enzymes but leave stable molecules intact. The other was kept cold, preserving the enzymes but eventually allowing the light-producing substrate to be used up. Separately, each extract eventually stopped glowing. But when Dubois mixed them together, the light returned. From this, he deduced that there must be two components, a substance that produced the light, which he called luciferin, from the Latin for light-bearer, and an enzyme that catalyzed the reaction, which he called luciferase. These names have stayed with us for more than a century, but he had identified the essential architecture of the system. The story moves again, now to the middle of the 20th century, and to a Japanese marine biologist named Osamu Shimomura. In 1955, Shimomura began studying a small and unremarkable-looking jellyfish called Equirria victoria, the crystal jellyfish found along the Pacific coast of North America. The jellyfish had a faint greenish glow around the edges of its bell. Shimomura wanted to understand where it came from. What followed was decades of patient work. Shimomura and his collaborators collected enormous numbers of these jellyfish, cutting away the glowing ring tissue and processing it carefully. From this tissue, he isolated a protein he called equirin, which emitted blue light in the presence of calcium ions, even without oxygen. This was already unusual. And then he found something else. The jellyfish does not actually glow blue, it glows green. Shimomura discovered a second protein in the tissue that absorbed the blue light from equirin and re-emitted it as green light. He called this protein green fluorescent protein, GFP. At the time, this seemed like an interesting detail in the biology of one particular jellyfish. It turned out to be one of the most important discoveries in the history of biological research. In the 1990s, other scientists realized that if you could attach the gene that codes for GFP to any other gene you were interested in, the cell would make that protein and it would glow green. You could watch it in a living cell. In real time, you could see exactly where a protein was, when it was being made, and how it moved. You could tag a cancer cell and watch it divide. You could mark a neuron and trace where it sent its signals. The ability to see inside living cells, to watch biology happening rather than just inferring it from dead tissue, transformed the life sciences. Shimomura, along with Martin Chalfie and Roger Tien, received the Nobel Prize in Chemistry in 2008 for this work. The jellyfish that had glowed quietly in the Pacific for millions of years had, without knowing it, been carrying a tool that would one day help us understand the living world from the inside. There is something gently moving about that. A small, transparent creature drifting in cold water, containing within itself a molecule that would one day illuminate the interiors of living cells, help us track the spread of disease, and reveal the workings of the brain. It did not know. It simply glowed. The science of bioluminescence is still, in many ways, a science of open questions. The more carefully researchers look, the more they find that they do not yet understand. We know, for instance, that the deep ocean glows, but we have explored only a small fraction of it. The creatures living in the greatest depths, at pressures that would crush any submarine we have yet built, remain largely unknown to us. Some of them almost certainly produce light in ways we have not yet described, using chemical pathways we have not yet identified. We know that bioluminescence has evolved at least 94 times, but we do not fully understand why it is so common in the ocean and so rare in fresh water. Rivers and lakes cover a significant portion of the Earth's surface. They contain an extraordinary diversity of life. And yet almost nothing in them glows. The reasons for this asymmetry are still being worked out, still being gently argued over in the patient way of scientists who have more questions than answers. We know that most bioluminescent light in the ocean is blue or blue-green because those wavelengths travel farthest through water. But some creatures produce red light, or orange, or even violet, and the functions of these rarer colors remain largely unknown. The stoplight loosejaw uses red light as a private beam, but that is one species among thousands. What the others are doing with their unusual colors, and why those colors were selected for rather than the more common blue, is still a question without a complete answer. And there is the question of borrowed light. This suggests that the ability to produce light can be distributed through food webs in ways that are still being traced, still being mapped. In April of 2023, researchers at Stanford published work describing a bioluminescent indicator engineered in the laboratory that glows when a cancer drug is active inside the brain. The ability to watch a drug working in real time, inside living tissue, without surgery, without removing samples, is the kind of development that changes how medicine is practiced. It came from understanding, deeply and precisely, how a tiny marine organism converts chemistry into light. And just recently, in 2026, researchers at the University of Colorado found a way to sustain the glow of a bioluminescent algae called Pyrocystis lunula for longer periods than had previously been possible. They embedded the living algae in a hydrogel, a soft material derived from natural sources, and identified the chemical stimulus that kept the light going. The eventual applications they imagine include streetlights that do not require electricity, living light sources that also capture carbon from the air, and illumination systems for autonomous robots exploring places where batteries cannot easily be replaced. The deep sea. It is worth pausing on that last image. A robot moving through the dark ocean or across the surface of some distant moon lit by the same chemistry that a firefly uses on a summer evening. The same molecules that Raphael Dubois isolated from his glowing clams more than a century ago. The same cold light that Aristotle noticed and named 2,000 years before that. Light without heat. Life illuminating the dark, patient and unhurried, the way it always has. Consider too what happens in your own body when darkness comes. You carry your own version of this relationship with light, written not in chemistry but in rhythm. As the evening deepens and the light fades, something in your brain registers the change. A small gland, tucked deep in the center of the skull, begins its slow work. Melatonin, the molecule of night, begins to rise in the bloodstream. Your temperature drops slightly. Your heart rate slows. The day's alertness begins to dissolve at its edges. This is not something you do. It is something that happens to you, the way the tide happens to the shore. Your body has been calibrated over millions of years to the rhythm of light and dark, to the turning of the planet, to the same ancient cycle that governs the bioluminescent bay and the firefly field and the glowing deep. You are part of that rhythm. You have always been part of it. The screens and the bright rooms and the artificial evenings of modern life have made it easier to forget. But the rhythm is still there, patient and deep, waiting for you to return to it. And every night when you finally lie down in the dark, when the light goes out and the room goes quiet, your body recognizes what is happening and begins, without any instruction from you, to do what it has always done. It prepares for the long, slow drift into sleep. The ocean at night, if you could see it from above, would look like a dark field scattered with moving lights. A ship passing through warm tropical waters will sometimes leave a glowing wake that stretches for kilometers behind it. Dinoflagellates disturbed by the passage, each one flashing its tiny blue alarm. Millions of them together producing a trail of cold fire on the surface of the sea. Those dinoflagellates, the same tiny organisms that make Mosquito Bay glow when you trail your hand through the water, use their light as a defense. When something brushes against them, they flash. The flash attracts attention, but not the attention of the thing that disturbed them. It attracts the attention of something larger, something that might eat the creature that disturbed the dinoflagellate. The light is a call for help. A burglar alarm, as some researchers have described it. A way of saying, in the only language available, something is here that should not be. There is a certain quiet humor in the image. A creature smaller than a grain of sand, invisible to the naked eye, defending itself against a copepod or a small fish by essentially shouting for a bigger predator to come and deal with the situation. And it works. The flash changes the dynamic. The predator, suddenly visible, suddenly exposed, often retreats. The dinoflagellate drifts on, dark again, unharmed. Somewhere in the shallows of a warm bay, a shark is moving. Not the large, familiar sharks of the open ocean, but a smaller species called the swell shark, found along the Pacific coast of North America. In daylight, it is a mottled brown, patterned for camouflage against the rocky sea floor. But under blue light, which is the kind of light that penetrates to the depths where swell sharks live, its skin glows green. Brightly. Distinctly. In a pattern that is specific to the species. The sharks can see this glow. Their eyes are sensitive to the wavelengths involved. Other species of shark, swimming nearby, cannot. The green glow is a private signal, a conversation conducted in a frequency that only the intended audience can receive. What the conversation says, what information is being exchanged, is still being studied. But the fact that these animals are carrying on a visual dialogue that is entirely invisible to human eyes without special equipment is a reminder that the world is full of communications we are not equipped to overhear. Bioluminescence, in this sense, is a window into a different way of being in the world. A way of perceiving and signaling and navigating that does not depend on the sun, that does not borrow light from anywhere external, that generates its own illumination from within. It is a kind of self-sufficiency, so complete and so elegant that it took us centuries to properly understand it, and we are still learning. The swell shark glows green. The hatchetfish erases its own shadow. The brittle star releases a glowing arm and walks away. The anglerfish dangles its living lantern in the dark. The dinoflagellate flashes its tiny alarm. The firefly writes its message in pulses of yellow-green light on a warm summer evening. The crystal jellyfish drifts in cold Pacific water, carrying in its tissues a protein that would one day illuminate the interiors of living cells. All of them making light. All of them using it differently. All of them part of a phenomenon so widespread, so ancient, so deeply woven into the fabric of life on this planet that it has evolved independently nearly 100 times and is still evolving, still finding new forms, still discovering new uses for the oldest trick in biology. There is something deeply restful in that continuity. Something that puts the day's concerns in a gentler perspective. Whatever you are carrying tonight, whatever is still unresolved, whatever felt heavy or complicated or too large to hold, it exists inside a world that has been quietly glowing in the dark for 500 million years. The ocean floors are lit by living creatures. The summer fields pulse with fireflies. The forests harbor their patient, steady fungi. The deep water carries its 80% of glowing things, going about their ancient business, entirely unaware of being observed. The world does not need your attention to keep going. It will glow whether you are watching or not. And so we leave it there. Somewhere below the surface of a dark, warm sea, something is making light. something small and patient and ancient, converting chemistry into photons with a quiet efficiency that no human technology has ever quite matched. The images will settle where they settle. The science will drift where it drifts. What remains, if anything does, might simply be the feeling of having spent some time in the company of a world that glows in the dark, without effort, without anxiety, without needing anything to be different from what it is. The day is behind you now. The night is doing what nights do. Sleep well. There is a creature called the vampire squid, and despite its name, it is neither a true squid nor a true octopus, but something in between, a remnant of an ancient lineage that has been drifting through the deep ocean for roughly 300 million years. It lives at depths where the pressure is immense and the temperature hovers just above freezing. Its body is dark red, almost black, and it wears a cloak of webbing between its arms that it can pull up over itself like a cape when threatened. The name, which translates from the Latin as vampire squid from hell, was given by a scientist who apparently felt that the occasion called for something memorable. When disturbed, the vampire squid does something that most cephalopods do not. It does not produce ink. At those depths, ink would be useless because there is no light for it to obscure. Instead, it produces light. It releases a cloud of bioluminescent mucus from the tips of its arms, a glowing, slowly dispersing cloud that hangs in the water where the animal was a moment ago. The predator is left investigating a fading smear of cold blue light while the vampire squid retreats into the dark. It is ink, reinvented for a world without sunlight. Further up in the water column, in the twilight zone where the last traces of daylight arrive as a dim blue suggestion, there are creatures called siphonophores. These are colonial organisms, which means that what looks like a single animal is actually a colony of many individuals, each one specialized for a different function. Some members of the colony are responsible for movement, some for feeding, some for reproduction, some for defense. Many siphonophores are bioluminescent. When disturbed, they can produce waves of blue light that travel along the length of the colony, a signal passing from one end to the other like a message moving down a wire. What that message means, whether it is coordinating a defensive response or simply a cascade effect with no particular meaning, is still being worked out. But watching footage of it, which researchers have captured with remotely operated vehicles in recent years, it is difficult not to feel that something is being communicated. The light moves with too much coherence to seem entirely accidental. There is a cave in New Zealand called Waitomo, and inside it, in the darkness of the limestone passages, the ceiling glows. Not from any geological process, not from minerals or reflected light, from larvae. The larvae of a species of fungus gnat called Arachnocampa luminosa, which exists nowhere else on Earth in quite this form, hang from the cave ceiling on threads of silk and produce a steady blue light from their tails. The light attracts small insects which become entangled in the silk threads. The larva reels them in and feeds. The effect, when you are standing in a boat on the underground river and looking up, is of a sky full of stars. A perfect, still, silent sky, blue-white and dense with points of light, reflected in the dark water below you. People who have seen it often reach for astronomical language without thinking. It looks like the Milky Way. It looks like the night sky on a mountain far from any city. It looks like something that should not be underground, in a cave produced by the hungry larvae of a small fly. The Maori people of New Zealand knew about the glowworms long before any European visitor arrived. They called them titiwai, which translates roughly as lights reflected in water. The name suggests that someone stood where visitors stand today on the dark underground river and looked up at the glowing ceiling and then down at its reflection, and understood that what they were seeing was a kind of doubling. Light above and light below, the cave holding both. Outside of caves, the land offers another encounter with bioluminescence that is less celebrated than fireflies, but no less strange. Railroad worms are the larvae of a beetle found in South America, and they glow in two colors simultaneously. Along their sides, they produce green-yellow light from a row of small organs. From their heads, they produce red light. The effect, when one is moving through leaf litter on a dark night, is of a tiny illuminated train moving through the undergrowth, which is presumably how they acquired their common name. Why they produce two colors is not fully understood. The red headlight may function as a warning, since many predators associate red with unpleasant experiences. The green body lights may serve a different purpose entirely. They are doing two things at once, in two different colors, and we are still working out what both of those things are. There is another scientist whose story belongs in this chapter, and whose path to discovery was considerably less straightforward than Shimomura's. He wrote a book called Bioluminescence, published in 1952, that ran to 649 pages and remained the definitive reference in the field for decades. Harvey's approach was methodical to the point of stubbornness. He traveled to collect specimens. He set up experiments in temporary laboratories in coastal towns. He corresponded with naturalists and fishermen and sailors who had reported strange glowing things in the water or on the shore. One story Harvey collected came from soldiers who had fought in the First World War, and it is one of the stranger intersections of bioluminescence and human history. In the muddy trenches of the Western Front, soldiers had noticed that wood taken from the shattered forests sometimes glowed in the dark. The glowing wood, colonized by bioluminescent fungi, was occasionally used by soldiers who needed just enough light to read a map without giving away their position. Not enough light to read comfortably. Barely enough to make out the lines and markings, but in conditions where any other light source would have been dangerous, the faint cold glow of a piece of foxfire was sometimes the only thing available. Harvey found this detail worth recording. Not because it was scientifically important, but because it illustrated something he believed deeply, that bioluminescence was not merely a laboratory curiosity, but a phenomenon woven into human experience in ways that were easy to overlook. People had been noticing it and using it and wondering about it for as long as there had been people to notice things in the dark. The thread continues into the present day and into medicine specifically. The luciferase enzyme, isolated and studied and eventually synthesized, has become one of the most widely used tools in biological research. When scientists want to know whether a particular gene is being expressed in a cell, whether a particular biological pathway is active, they can attach the luciferase gene to whatever they are studying. If the process is happening, the cell glows. If it is not, the cell stays dark. The presence or absence of light becomes a direct readout of biological activity. This has been used to track the replication of viruses inside living organisms, to watch tumors grow and shrink in response to treatment, to study the circadian rhythms of individual cells, and to map the spread of infection through tissues in real time. During the development of vaccines and antiviral drugs, luciferase assays, as these experiments are called, have been among the most widely used tools for testing whether a compound is doing what it is supposed to do. The molecule that makes a firefly flash has been put to work in the service of human health, and it has been extraordinarily useful. There is a particular experiment that researchers at the Salk Institute conducted in the early 2000s that deserves a moment of quiet attention. They were studying the master clock of the circadian system, a small cluster of neurons in the brain called the suprachiasmatic nucleus, which is responsible for coordinating the body's daily rhythms. They engineered mice in which the cells of this clock expressed a bioluminescent protein whenever the clock genes were active. Then they placed a very sensitive camera over the brain and simply watched. What they saw was a wave of light, moving through the tiny cluster of neurons in a 24-hour rhythm as reliable as the turning of the earth. The clock was visible, not inferred from behavior, not calculated from blood samples, but actually visible, a living glow cycling through the brain of a living animal, marking the hours, the light of a jellyfish, adapted and repurposed, revealing the hidden timekeeper inside a mammalian brain. It is a long way from the cold Pacific waters where Equaria Victoria drifts, and yet the connection is direct, traceable, a single thread running from a transparent jellyfish to a Nobel Prize to a glowing brain. Science, at its best, has this quality of unexpected connection, of finding that the answer to one question was hidden inside a creature that had nothing obvious to do with the question being asked. There are people who have made it their life's work to seek out bioluminescence in places where it has not yet been described. Marine biologists who design expeditions around the goal of descending to depths that have never been visited and simply watching what happens when the lights of the submersible are turned off. The protocol is straightforward in description and extraordinary in practice. You descend in a small vessel to a depth of perhaps 500 meters. You turn off all your own lights. You wait for your eyes to adjust, which takes longer in complete darkness than most people expect. And then you watch. What researchers have reported from these dark adapted dives is a world that is far more luminous than anyone anticipated. Flashes and glows and trails of light moving in every direction. Large animals passing in the distance and leaving brief smears of cold blue behind them. Smaller creatures producing rapid rhythmic pulses that suggest signaling rather than defense. The impression, described by more than one researcher, is of a city seen from a great height at night. Points of light, moving lights, lights that appear and fade, an entire landscape of illumination that exists without any input from the sun. Edith Widder is one of the scientists who has spent the most time in this dark world. She is a marine biologist who has made more than 200 dives and submersibles, and who developed a camera system specifically designed to detect and record bioluminescence at depth. One of her most significant discoveries came from an unexpected direction. She and her colleagues designed a device called an electronic jellyfish, a ring of blue LEDs programmed to mimic the bioluminescent distress signal of a dinoflagellate being eaten. The logic was that in the deep ocean, a small creature flashing an alarm might attract a larger predator. So if you mimicked the alarm, you might attract the predator. They deployed the device at 600 meters and watched what came to investigate. Among the visitors was a squid so large and so rarely observed that it had previously been known only from dead specimens, from beaks found in the stomachs of sperm whales, from the occasional carcass washed up on a remote beach. Architeuthis, Dews, the giant squid. It came to the electronic jellyfish, reached out its long tentacles, and was filmed for the first time in its natural habitat. The footage, when it was finally released, was watched by millions of people who had grown up reading about the giant squid as a creature of legend, something that was known to exist but had never been properly seen. It was found, in the end, by light, by the mimicry of a tiny organism's distress signal broadcast into the dark water at 600 meters, answered by something ancient and enormous that had been living in that darkness all along. The question of why so much of the ocean glows while fresh water remains almost entirely dark has led researchers towards some interesting hypotheses. One possibility involves salt. The ionic environment of seawater is different from freshwater in ways that may make certain bioluminescent chemical reactions more stable or more efficient. Another possibility involves the history of life itself. The deep ocean has been continuously inhabited for far longer than freshwater bodies, which have been periodically disrupted by geological events, by the freezing and thawing of ice ages, by the formation and disappearance of lakes and rivers over geological time. The ocean has had more time to develop and refine its bioluminescent systems. Freshwater has been, in evolutionary terms, a more interrupted and unpredictable environment. A third possibility, and perhaps the most intriguing, involves predation pressure. The deep ocean is a place where being seen can mean being eaten, and where producing light is therefore both a risk and a necessity. The pressure to evolve bioluminescent defenses, lures, camouflage, and signals may simply be much higher in the ocean than in freshwater, where visual predation works differently and where the distances involved are much smaller. In a lake, you do not need to signal across kilometers of dark water. In the deep ocean, you do. These are still hypotheses, still being tested, still being debated in the careful, patient way of scientists who understand that the ocean is very large and that certainty arrives slowly. But the questions themselves have a kind of beauty. They point toward the deep interconnection between the physical conditions of an environment and the biology that develops within it. The ocean glows because the ocean is the kind of place that rewards glowing. Life found the light, and the light found its uses, and the two have been refining each other for 500 million years. Outside the ocean and the forest, in a place that most people would not think to look, bioluminescence has recently been found in a group of animals that surprised nearly everyone who studies the phenomenon. Sharks, not just the swell shark with its green glow, but several other species, including the chain cat shark and the small spotted cat shark, which were found in 2021 to produce bright green bioluminescence across their bodies in patterns that are entirely invisible in white light. The patterns are complex and species-specific. They vary between males and females. They are brightest on the fins and along the flanks, in places that would be visible to another shark approaching from the side. The researchers who described this finding noted that the shark's eyes are well adapted to detect the specific wavelengths involved and that the patterns might function in species recognition, in communication between individuals, or in behaviors related to finding a mate. The sharks have been carrying on a conversation in green light in the shallow reefs where they live for an unknown length of time. We only recently became aware of it because someone thought to look with the right kind of camera. It is a small lesson in the limits of what we know. The world is not only stranger than we imagine, it is stranger than we can easily hold in mind at any one time. While we are busy with our days, with the conversations and the worries and the small urgencies that fill the hours, the sharks are glowing green in the shallows. The fungi are patiently illuminating the forest floor. The dinoflagellates are flashing their tiny alarms in the warm, dark water of a bay in Puerto Rico. The deep ocean is doing what it has always done, lit from within by the creatures that live there, unhurried and ancient and entirely unaware of being remarkable.